Thank you very much. I'm going to assume everyone can hear me, but let me know in the chat if you cannot. Right, so I'm very honored to be here. My name is Lukasz Bugaj. I am at the University of Pennsylvania in bioengineering. We started our lab about 5 years ago, and very fascinated by all the topics in this really wonderful symposium. Sorry I couldn't be there in person. We had some childcare issues we couldn't resolve, and I'm hoping that they will. My children will let me complete this without interruption, but we'll see. What we are interested in our lab is to understand cell signal perception, right? And even beyond that, misperception. So generally, we're interested in knowing how cells process inputs, process their environments through signaling networks to make the appropriate cell-fate decision, right? And to this audience, I think I don't have to actually spend much time talking about how signaling networks are really the brains of the cell, right? Just like in our visual system, when the photon hits your eyeball, it doesn't really mean anything. That's just the first step until that sensation is really processed, perceived by the brain, that then allows you to make an appropriate response. So cells really have the same, are doing the same thing. So cells are studded with antenna or receptors that bind biochemical information, and again, that sensing of the ligands in the environment is just the first step. But then that's transduced into this complex activation of many signals inside the cell, and as a result of which the cell then interprets or perceives that external information and then makes the appropriate response, right? So if you can accept that a cell can perceive, then it's only one step further to think maybe if a cell could also misperceive. And so we're interested in understanding cell signaling from this alternative perspective. And one of the things that we sort of our approach to this is by developing really precise molecular tools to allow us to ask these sorts of questions. Actually, very similar to the same sorts of tools that were developed for neurons to understand brains by using the precision of light control. We're actually using those same types of approaches to sort of reach into the cell on a much smaller level and have precise control in space and time over individual nodes of signaling networks to understand how they control cell function. So I'll give you a very brief introduction to our lab and how we approach this. Again, half of what we do is the molecular engineering to make the proteins that allow us to do this sort of work. So these are optogenetic proteins, light-sensitive proteins that you can fuse to your favorite signaling protein in a cell, express it, the cell makes the protein, and then just by shining light on the cell, you can control the activation or inactivation of that particular signal. We do a lot of engineering on the protein side. We do some hardware engineering also to allow us to actually apply lights in this manner, actually in high throughput, also to give you sort of arbitrary control in hundreds of experiments miniaturized at the same time. These tools are actually going to be featured throughout our talk. We are also developing other sorts of approaches now, moving beyond light to use temperature to control proteins inside cells with similar precision. The main goal there being sort of when you want to do these sorts of studies, but maybe in an actual animal where light doesn't penetrate, temperature is a better penetrant dynamic stimulus. We do some biosensor design to look at some molecular features like protein clustering that, again, won't really feature in this talk. But then we apply these sorts of tools that we develop and that others have developed to really understand how our signals process in cells and how can that go wrong, particularly in the context of cancer. And that's where really the talk is going to focus today, is on cell signaling in cancer. But I do want to start briefly with a couple slides just on what optogenetics is. Many of you, particularly those more familiar with the neurosciences, will be familiar with channel rhodopsins, but this is a slightly different different set of tools, but under the same name. So as a brief introduction, obviously many types of organisms have light responsive elements and behaviors. Plants maybe come to mind first for many people. And so these sorts of, for example, growing towards light, that is often mediated by proteins inside the cells of that plant that themselves respond to light. Okay? So we can take the genetic code from those proteins. Often they respond to blue light or red light. And if we do it right, and if we attach it to our signals of interest, we can then control those signals inside mammalian cells with those same light inputs. Okay, there are now many ways of doing this. This field is, well, depending from where you start counting, but at least 15 years old. One example, this clustering protein that I showed you before is near and dear to my heart. This was from my graduate work, where we really found this naturally existing a protein from Arabidopsis called Cry2 that when you shine light on it, it clusters. You take blue light away, and then the clusters diffuse back into the cytoplasm. This is in mammalian cells now. With very little optimization, this works great in mammalian cells. And this can actually this is basically this modular thing that clusters in response to light, and you can apply it to activate so many things inside of a cell. Turns out clustering is a really powerful form of protein actuation. In our case, we were interested in a signal pathway called beta-catenin to understand how neural stem cells differentiate. You can see whenever the light's on, the transcription of the pathway's on. You turn the light off, the transcription turns off. You can study dynamics of signaling and how that controls stem cell differentiation into mature neurons. You can look at more fast timescale things like cell migration. So we can use this to control a cell's own receptors that control cell migration. And now wherever we paint the cell with light, the cell now moves in that direction, and we can sort of make it go wherever we want. Okay. Many other applications just for this kind of clustering approach, but also many other types of ways to do optogenetics inside of a cell, again, that are not ion channels. So you can, for example, use dimerization to recruit proteins to the plasma membrane where a lot of signals are started, or you can change conformation specifically of certain proteins just with light inputs. Okay, there's a whole tool set that's actually quite actually kind of complex though if you're not really uninitiated, but that basically allows you to do any number of things. I think kind of anything, any kind of molecular actuation you want to do inside of a cell, there's a way to do it optically now. Okay, so that's my brief intro, and now I really want to tell you what are the types of things that we're doing with this tool set, particularly in helping understand the complexity and the signal processing of cells in health and in disease. Okay, and so particularly in the context of cancer, the sort of the big idea here is to take just a different approach to understanding how cells are functioning and how cancer cells are malfunctioning. And so because we have these really precise optical inputs, with light that we can plug in sort of anywhere in a pathway, and we have really precise outputs, for example, live cell biosensors or transcriptional responses, or just watching how cells differentiate. We can actually really treat the cell like a black box, right? And so we can say, much like an electrical engineer would characterize a complex circuit by giving a precise impulse and seeing how that's transformed by the circuit as an output. We can do the same thing now, and to understand the function of cells through these signaling pathways. This is work that really started when I was a postdoc at UCSF with Wendell Lim, and this was really just a hypothesis at that point, right? So we knew we had these tools, but so the question was, can we first quantify signal processing, and then can we actually measure changes that are meaningful in the context of cancer? And so we started with cancers that are mutated in this particular pathway called RAS-ERK. This is an ancient pathway involved in growth and proliferation and differentiation. So really kind of any essential cell process, and often mutated, and because it's so central, it's mutated in half of human cancer. So it's super important clinically, and particularly in lung cancer, which is where we started looking. Okay, and so we actually had a small panel of cancer cell lines where we expressed this protein called optosas. This is this activator of this top enzyme called RAS, and this is basically how these experiments look. So we could, by shining light on the cell, we recruited optosas to the membrane, and that's where the signaling is initiated in this pathway. And then downstream, we had a fluorescent reporter of ERK activity. Okay? And we just looked at the input-output response. And we found, the first thing we found really was this entire class of cancer cells where again, this input-output response was really kind of significantly altered, right? And it looked this way. And the phenotype was a dynamic one, right? So in most normal cells, or actually all normal cells that we looked at, like these fibroblasts, if you give a step input at the top level of the pathway and you look downstream, this ERK activity follows very rapidly and very closely to your RAS inputs, okay? So within a couple minutes of activating of RAS, we get this really sharp activation of ERK, and then with inactivation, you get a very sharp inactivation. But again, there was this whole class of cancer cells where these transitions were actually extended, right? So you had a much slower ramp to turn on, but particularly even more so, a much slower ramp to turn off once you remove this very precisely this optical signal. So the easiest way to really interpret this is that the area under the curve. So in response to the same input, the area under the curve now is different, right? The cell's perceiving a sort of a different external input than is actually there. And you can imagine that there will be a certain input, a dynamic input space where this pulsatile input, where a normal cell would perceive these input pulses downstream of the pathway still as pulsatile, these cancer cells, because of these extended kinetics, will now blend those pulses and perceive it as a constant input, okay? And so these pulses versus a constant signal can now mean very different things for the cell. And we could hypothesize this, that there was maybe one particular signal, but we could also, with modeling, but also with our high throughput optogenetics approach, we could now just sample the entire a very large space of dynamic inputs where we can systematically modulate the pulses and the pulse intervals, and show that for these cells with extended kinetics, you now have a much larger space of inputs that the cells would now proliferate in response to. So really, our first example, yes, actually, in the context of disease, you could get this misperception, particularly of these dynamic pulsatile inputs. And just another way to think about this is, this oncogene for some reason has now given this whole, kind of a blurred understanding of what's actually, outside, where these low frequency signals, yeah, you can still sort of, make them out. But now when you start to get to more higher frequency, finer resolved differences, those start to be, possibly confused. So again, that was really the first proof of principle that this was there was something there to look into. And when I started my own lab again about 5 years ago, one of our big questions was, what else is out there? What else could we possibly find? You could imagine many different types of misperception in a disease state. And so given our history, especially in modulating clusters and thinking about protein clustering, we were very drawn to this particular different class of cancers, also lung cancers, driven by an oncogene called EML4-ALK. And it's depicted here in green. And the first thing you just kind of hits you, it's kind of impossible to ignore is its localization, these aggregates that form in the cytoplasm. Okay, and I'll get back to these aggregates in a second. But let me tell you a little bit about this oncogene. It's a little bit different from most of the ones that you may have heard of that are just kind of point mutants. These are called RTK fusions, or receptor tyrosine kinase fusions, and they form not through point mutations, but actually through rearrangements of chromosomes or inversions of chromosomes that, as a result, make these sort of hybrid chimeric proteins that are the intracellular or the business end of a receptor tyrosine kinase, where the enzymatic domain is, and it's fused to some partner protein from some other protein in the cell, right? And the thing that these partners often have in common is that they're often oligomeric. There's many of these. There's over 1,000 that have been, characterized. Actually, not even characterized, but found. And some estimates put them up to, present in over, about 5% of all cancers, which is the highest estimate I've seen. But they are, depending on the different cancer type and the different fusion, the numbers vary. The one that we're looking at is called, again, EML4-ALK. So this is the ALK kinase intracellular domain fused to this EML4. This is a microtubule associated protein. It's only the oligomerizing domain again, and it's fused to this ALK kinase. Okay, so this protein again shows up mostly in lung cancer. It's the deadliest cancer in the U.S. It activates many signals like a normal receptor would, but the most important one for oncogenesis is thought to be the same RAS-ERK pathway that we talked about just a couple of slides ago. There are great drugs that block the activity of ALK, but like all targeted therapies, resistance ultimately emerges. So really highlighting this continued need that it's not enough to just have tight binding drugs. We really need to better understand how the oncogenes, the host cell, and these drugs are all interacting to both kill cells, but also to allow cells to not die and to gain resistance. So again, this is really one of the main challenges in targeted therapy. And what I see, there are many ways that cancer cells can become resistant, but in a nutshell, sort of a big picture, you have cancer cells that are proliferating. If you inhibit this oncogene, in many cases, many of these cells will die. Most of the cells will die. In fact, almost all of the cells will die through a process known as oncogene addiction, because you've taken away this strong oncogenic signal. But the problem is that not all of them die, and even a small fraction, 1 in 1,000, 1 in 10,000 survive. They find a way to persist long enough to gain beneficial mutations that allows them to develop stable resistance that then ultimately allows them to regrow and repopulate a tumor, and that's tumor recurrence. So the thought is that if we can actually make this process more efficient, for example, if we can figure out why cells aren't dying, or if we can kill them faster, this could forestall or even prevent resistance in many cases. So, okay, so that's the sort of the big picture, but why, from a very basic fundamental signaling perspective, we were drawn to this is because, we're thinking, well, how can oncogenes maybe mess with the proper, transfer function, the proper perception of signals? These cancer cells have a big task, because they have two sources of these very important transmembrane signal, or I should say, two sources of the same signals. One is this normal transmembrane receptor that, again, senses environmental ligands, brings that into the cell, and allows the cell to perceive what it should perceive. But now they also have these gigantic aggregates that are signaling through the exact same pathways using the exact same proteins, right? They have these adapter proteins that couple the receptors to these, let's say the RAS-ERK pathway, which is what we're going to focus on. So, we just wanted to know what effect is there, if any, of these sorts of oncogenes? And you could hand wave your way to kind of any answer. Maybe they're synergistic, maybe they antagonize each other, maybe they're compartmentalized and they don't, but we didn't know. But we knew it was important because of the sort of the central role of this pathway, both in cancer and in drug resistance. So I started with optogenetics, and actually turns out that answering these types of questions is very complicated because of the how feedback and connected all of these biochemical pathways are. But optogenetics gives us this a very nice scalpel to get at some of this. So there's three main areas where we used optogenetics, and they're all different. So I'll take my time to sort of walk through them. This is the first. So the first was the very exploratory. What happens when we activate RTKs? And so instead of relying on the cell's own RTKs, we come in with our own receptor. And this is an optogenetic FGFR. So a different kind of, one kind of RTK that exists in cells, activated by the same optogenetic clustering molecule that I mentioned earlier, this Cry2. Turns out just by clustering this receptor molecule, you can activate this receptor strongly and then activate this pathway. And then we can look at what happens downstream. Now we won't do live cell imaging, we'll do just immunofluorescence for phosphorylation. And that's the active state. Okay, and this is really the kind of the central result, which is actually a negative result, but underlies everything I'm going to tell you about. So what we found when we turned the light on is that this is single cell immunofluorescence, and you can see basically the light gave us this tiny shift in the distribution of activated signal. Okay, this is basically no response. And we were very surprised by this because these optogenetic tools are very, very strong. And so we expected a much stronger signal, but we saw basically none. And the first response is always, well, maybe this is a cancer cell. It already has a lot of high signaling because that's what the oncogene does. And so why would you expect a signal? But actually that's not the case, because when you do this experiment in the presence now of an ALK inhibitor, two things happen. One is that the basal signal goes down, and that makes sense because you block the oncogene. But now you turn the light on, and now you can drive signal, 10, 20-fold through the same pathway, same cells, same light input, but just now that you have the drug there. And not only can you does the fold change of the signal increase, but you can always drive it to a higher maximal level than you ever could in the absence of drug. So the drug, it's supposed to block signaling, but it's actually increasing your potential to signal through the pathway when you stimulate it with a membrane receptor. This is just imaging of the immunofluorescence. Here with drug, you're just seeing a brighter signal than without drug. Okay, so what this says is that the activity of the oncogene is actually somehow suppressing transmembrane receptor sensitivity. This was unexpected. So we try to actually, before I can show you this again, we're gonna you can see this through a couple different methods. First was optogenetics. This is actually now just, you can add the ligand of a receptor in these cells already, so nothing synthetic about this. And this is now a live cell reporter of ERK activity. So this reporter is cytoplasmic when the pathway is on, and it's nuclear when the pathway is off. So this is here cells in the absence of a drug. The fluorescence is a little bit in the nucleus, a little bit in the cytoplasm. And what we're going to do here is we're going to add ligand when I press play. And what you'll see here is that you'll see fluorescence come out of the nucleus, which means the pathway is turning on. What you'll see here in the presence of drug, it does so much stronger. There's much more nuclear exclusion than in the absence of the drug. Okay, so again, the drug is potentiating signaling going through this pathway. Somewhat paradoxical. Okay, and this is quantification showing that we can show actual sensitization to about like an order of magnitude lower concentrations when you add the drug than in the absence of the drug. Okay, so the second place optogenetics really helped us is now that we know there's some effect of this oncogene, the question is, where is it? And there are, again, many connections, many papers showing different connections that you have to test out. But one thing optogenetics does, it's allow us to also kind of narrow down the search space. So because you could have these interactions at any number of nodes, one thing you could do is you can have multiple different probes that can allow you to kind of walk down the pathway and ask, well, what if I activate, at the level of RAS or at the level of RAF? Do you still see the phenotype? And if you do, that means that, whatever interaction you were looking for is further downstream. And so you can keep going down. Okay. In our case, we only had to do 2 of these. This is the activating the receptor level, which I just showed you. So here, in the absence of drug, you get a small response. In the presence of drug, you get a very large response, again, larger than you did without the drug. But if we do the same experiment, but now activating 1 step downstream, activating at the level of RAS, now you see there's no dependence on the drug. Doesn't matter if the drug is there, you can activate to the same maximal level. Okay. What this tells us is that actually it dramatically narrows down our search space. It tells us whatever we're looking for is between the level of receptor activation and RAS activation. Okay. And it looks like there's not much there, but actually there's one very important level. And before I get to that, the third area where we can really, this gets a little next level here in terms of like complexity, but there's an entire literature of other cancers where similar phenotypes have been found, and they've been attributed to this negative feedback. So ERK activates transcription and proliferation, but also suppresses receptors through a number of different mechanisms that have been found, right? Each of which kind of requires its own assay and characterization. But here again, we could also test them all at once. So the issue here is that we know that there may be some direct interaction, or we have some negative feedback that's coming from ERK. When you just add ALK inhibitor, you actually block everything, right? You block any direct interaction. You also block ERK activity, so you're losing this negative feedback. But what we could do is we could add ALK inhibitor and at the same time activate 1 step downstream from the receptor at RAS. Okay, so now we can sort of decouple ALK activity from ERK activity. We can maintain high negative feedback, but suppress any other interactions and see what happens, right? And so this is the result I just showed you 3 times, that in the presence of drug, you get a stronger response to EGF. Now, if I turn the light on and do the same experiment, if the signaling goes back down, that means there's some negative feedback coming from ERK. But that's not what happens. The signal stays high, okay? So what that means is that in our case, this negative feedback plays a very minor role, if any. Okay. So I showed you what doesn't happen. What does happen is the following is, this is the picture that I painted for you in the beginning, where we don't really know what's happening. They're kind of sharing resources, but we think actually the actual picture looks much more like this, where these aggregates act as a sponge really for all of these adapter proteins that couple receptor activity to downstream pathway activation. Okay? And we did this many different ways. I don't have that much time, but I think we've heard seeing is believing a couple of times here. And so I absolutely believe that as well. These are cells where we have the adapters, these that we're claiming are being sequestered, tagged at the endogenous locus with fluorescent protein. And so then we transiently transfect them with this oncogene, and it's very easy to see which cells got the plasmid and which didn't. But the cells that got the plasmid have these gigantic condensates of, this is adapter, but it localizes to the oncogene. And the cells where the adapters diffuse, no oncogene. Okay. I press play, we're going to add a ligand, which stimulates the pathway. Normally, in normal cells, the adapter will go to the membrane, which is where the receptor is. And that's what you see in these diffuse cells. I loop this for clarity so you can see this. It happens very fast. But you see this translocation in the normal cells, but in the cells where you see the condensates, which have the oncogene, no translocation. Okay, and we can quantify this. And you basically, here's the translocation without without oncogene. With oncogene, it basically goes down to 0. Okay, and so then we can do the same experiment, but do it in the presence of drug. And we can see that in the presence of drug, the adapters diffuse into the cytoplasm. And now you add EGF. Now in a cell that has oncogene, you get really nice membrane translocation. Okay, and you can quantify this. So many other assays to show the same point, but in effect, our model here is that that the oncogene dynamically suppresses very strongly the transmembrane receptors that by the sequestration of shared adapters. So even if you have the ligand, nothing really happens. You add ALK inhibitor, it blocks the oncogene, but it also redistributes all of these adapters, which now strongly potentiates receptor signaling through normal, unmutated transmembrane receptors. Okay, so we were happy to sort of figure out this molecular nature of how we can make cells restore perception of external signals. But the big question we had was, does this matter, right? So particularly in the case of drug treatment, is this actually useful for anything to do with cancer? And I hope to convince you that it is. And so at this point, we're going to forget optogenetics or external stimulation. We're just going to watch the cells with this same reporter as we treat the cells with drug. Okay. So I press play and we add the drug, and you see the fluorescence goes to the nucleus, which means that oncogene is turning off. Everything makes sense. But now, as you continue the movie, within about an hour or two, something very interesting happens, and you start to see these pulses, kind of sporadic throughout the population, not random, spatially coordinated, but definitely, I would call them sporadic. The other thing you see is that you see these fluorescent puncta. These puncta are actually dead cells. Okay, so we're adding an oncogene inhibitor. Oncogene addiction, in this case, says that some of these cells will die, and that these are the dead cells. So we're very surprised by this, but also very curious. So we dug a little further, and so what you can see is that actually these, I also bring up the death and the pulsing because they're very closely linked. These are stills from that same movie. You have a cell that's dying. Now it dies, its neighbor pulse, and then the pulses are very short, like 15 minutes. Okay. Without drugs, some cells die, but the neighbors don't pulse. Okay. And so this really tipped us off that maybe actually there's some cell-to-cell signaling happening during apoptosis, during death. And this has actually been well characterized for really over a couple decades, but in the past couple of years, very well done with looking at cells, movies, live cell biosensors. And the idea is that basically when an epithelial cell dies, it sheds EGF ligands, so growth factor ligands, to its neighbors to allow them to fill the hole that's resulting from a dying cell. And we thought that's what was happening here. And this is great because there are two really nice drugs that we could test to either block the receptor on the receiving cell or the sending MMP inhibitors block the sending of the ligand. Okay. And basically what we found is that co-treatment of ALK inhibitor plus either of these drugs blocks all of this pulsing, right? Here are cells dying, the neighbors no longer pulse in response to either of these co-therapies. Okay, we can do this very systematically. We can find every single cell that died on our video. We can identify neighbors in a certain radius and compare the pulsing in those neighbors to non-neighbors. And we can find that without drug, some cells die, but again, there's no pulsing. When we add ALK inhibitor, there's a lot of pulsing, and all of it is in these neighborhoods of dying cells. And then when we do these co-therapies, we can block this pulsing. Okay, so I'll just finish up here with a couple slides showing that actually this is quite important actually in cell death. So with these monotherapies of just ALK inhibitor, we see that cells die, but at a certain rate. Now, when you block this paracrine signal, you can actually speed the cell death within 1 day, even by blocking this paracrine signaling, and these results hold out to 3 weeks. After about 3 weeks, just the single therapy, you see a regrowth of cells. This is the first steps of drug tolerance and resistance. But both of these co-therapies, you don't see it. They do a much better job of clearing the cells. Okay, so this is a rehashing of the mechanism. I hear that I'm out of time, but also out of time. The last thing I'll say is that I think that we found this with a very particular oncogene that we studied very closely. But all of these mechanisms, from the sequestration to the paracrine signaling from cell death, are very general. And so here we're showing that EML4ALK actually suppresses the ability of the cell to actually sense its environment. And it's not the only cancer where this happens. Some of the ones I mentioned earlier, different cancers, different tissues, different molecular mechanism, but same end result where the receptors are suppressed. And there's evidence all the time of more similar behaviors. And so it actually really, makes you wonder if this is actually not just something to be, important for cell death and resistance, but also for oncogenesis itself and understanding. We're very interested to understand sort of, does this play some role in forming permissive conditions for cancer? So with that, I'll thank you for your attention and thank everyone involved. In particular, David Gonzalez-Martinez is a PhD student, and Lee Roth is a postdoc in my lab. So thank you very much.